Assalamualaikum and hi everyone. I'm Siti FATIMAH binti KAMARUZAMAN. We are from Group 3 are going to present our summary of the report. To speak, the information we are going to share comes from our experience. On our outline, we are going to discuss about the total suspended solid, turbidity, pH indicator, soil type, geological measurement, backyard biodiversity, and lastly, waste segregation. Without further, let's talk about total suspended solid. Total suspended solid is indeed the dry weight of non-dissolved suspended particle in a water sample that can be tracked by a filter evaluated using a filtration apparatus. So, how total suspended solid can be done at home? Based on our experience, we collected several types of water sample such as aquarium water, river water, and drinking water into 200 milliliter clear bottle. Next, we set up all the sample at the same place under sunlight and left them for 3 days. After 3 days, we use laboratory equipment to measure the total suspended solid in our water sample. So that's all from me. I will pass to Nadia, who will describe more about this.
Thank you, Fatima. Hello, everyone. My name is NADIAH binti ZAINAL ABIDIN. So today, I'll be talking about the result of the experiment. As for the result, we took 3 samples, which are river water, aquarium water, and mineral water. First, we take an initial weight each of the samples, and after 3 days, we weigh all of the sample and calculate the TSS by using total suspended solid formula, which is final weight minus initial weight and divided by the volume of the sample. As we can see here, for the river water, the total suspended solid obtained is 67.2, while aquarium sample is 10 and mineral water is 0. Next slide. As we can see from the result, we can conclude that river water has the highest total suspended solid compared to the other 2 samples, while mineral water recorded the lowest to none value of the total suspended solid. Based on DOE water quality classification, the river and aquarium water sample categories is in polluted index range. On the other hand, according to DOE water quality index classification parameter, the river sample is in class 3, while the aquarium water is sample in class 1. For the class 3, the water are considered dirty and it is not safe for water supply, and for class 1 is practically no treatment necessary, and it has a very sensitive aquatic species. I will pass the presentation to the next presenter.
Thank you, Nadia. My name is Siti AISHAH binti DAUD, and I will continue with the second lab practical, which is turbidity. The definition of turbidity is cloudiness or haziness of a fluid caused by suspended solid, and the highest turbidity can prevent the amount of light from reaching deeper depth in water, such as river and lakes. We make turbidity measurement using 3 types of water, which is rice wash water, milk, and drinking water as a blend. In this practical, we use the Chinese method to find the turbidity for 3 types of water. We also tried the Tyndall effect theory to support the turbidity with 3 different of water. As you can see in the slide. Next. And in this slide, this is a result for turbidity. And for the conclusion is rice wash water and milk were more than 5 MTU. So it means they have higher concentration of particle. And based on WHO guideline, drinking water should have a turbidity of less or equal than 5 MTU. And it must be colorless. So it means there were less particles suspended in the water. That's all for me. I will pass to the next presenter.
Hi, I'm SYAFIQA ALMIRA, and today I'll be presenting about Lab Practical 3 Homemade pH Indicator. What is pH indicator and anthocyanins? A pH indicator or acid-base indicator is a compound that changes color in solution over a narrow range of pH values. Anthocyanins are a major class of red to blue flavonoid pigments that are extensively represented in plants. To identify whether the food used is acid, alkaline, or neutral, we need to see the color changes after mixing with the indicator red cabbage. Next slide. What What are the techniques? The technique that we used was extracting the indicator by boiling the red cabbage for 15 minutes and strain the mixture. In order to measure how acidic or basic a liquid is, the pH scale was referred to. Next slide. What are the results of this experiment? For vinegar, the vinegar solution turns red. Vinegar is a strong acidic and the pH value is between 2 and 3. Meanwhile, for the capsicum juice solution turns green. Capsicum juice is weak bases and the pH value is between 6 to 8. Next slide. What is the conclusion of this experiment? In conclusion, for this experiment, we use red cabbage as an indicator and we can identify which food items are acid or alkaline. For food that contains acid-forming substances like vinegar, the solution turns to red, purple, and violet. Meanwhile, for food that contain alkaline-forming substances like capsicum juice, the solution turns to blue and green. That's all from me. Thank you. I will pass to the next presenter.
Okay, thank you, Amira, for giving me the floor. Hello, everyone. This is Sumaya, and I'll be talking about the soil type determination. Soil is one of the most important natural resources on Earth, and it is made up some small particles, water, living organisms, and many more things. And the texture and the type of the soil helps us to ensure the plant health. And for this lab report, we determine the soil type from our home. And for the procedure, we can see here first we selected 3 clean glass jars and we labeled them as type A, B, and C. After that, we collected some of the soils from our backyard. We digged the soil from at least 6 inches of depth and removed all the debris, and we mixed some soap water with it. And after mixing it correctly, we kept the jars for 3 days. After 3 days, we noticed that there were 3 layers, which is silt, clay, and sand. And we measured the height of the layers by a ruler. And as for the result, we use the soil texture triangle to find the result. And for the type A, we noticed that it had 29% of sand, 60% of silt, and 11% of clay. Thus, it was a silt loam kind of soil. As for type B, it had 61% of sand, 28% of silt, and 11% of clay. It was a sandy loam kind of soil. And for type C, it was a sandy loam kind of soil as well, because it had 74% of sand, 18% of silt, and 8% of clay. And for the conclusion of this report, we can say that sample A and B had the best combination of sand, silt, and clay for a plant to grow, because this combination helped to form a loam kind of soil. That's all for this lab report. I'll pass the floor to the next representative.
Hi, my name is NURUL binti OTHMAN, and I will talk about lab 5 geological measurement. So, geological measurement is the orientation of a planar feature such as a bed of sedimentary rock that can be described with 2 values. The first one is strike. Strike is the compass orientation of the horizontal line on the surface of the bed, and dip is the angle at which the surface tilts down from the horizontal. Next slide. So how to draw strike and dip? Firstly, draw the strike line with the correct orientation between 0 to 316 degrees. Secondly, use left hand law. The index finger is the strike and the thumb is the dip. And thirdly, record the amount of dip between 0 to 90 degrees at the tip of the dip line. Next slide. And for the strike and dip exercise, the first question is 14 degree strike and 15 degree dip. So we use our left hand law, and the strike is index finger is 14 degree and the dip is 15 degree. So the dip direction the below. So the second question is 45 strike and the dip is 73 degree. So the dip is also direct the below. So that's all from me. I will pass to the next presenter.
Okay, thank you. My name is GUOQIAN, and now we are going to the next part. It is backyard biodiversity determination. Our globe have many symbols like trees, flowers, keys, hamsters, and some insects. Next step. We want to determine appropriate biological field measurement techniques and the measurements of biological parameter in the field. Biodiversity is a term used to describe the enormous variety of life on Earth. It can be used more specifically to refer to all of the species in one region or ecosystem. There are two types of biological sampling methods. The first one is standardized methods, which are projects, transect length, capture, and release. The second one is informal methods, which are direct observation of the animal insect, songs, and vocalization, scat, tracks, and trails, nests, scratching and rubbings, noise, and lastly logics. Thank you. This is all my part, and please the next presenter.
Thank you, Guoqian. Hello, everyone. My name is Erika, and I will be representing the result of this experiment. The biodiversity index quantifies the number of the different species in a given community and considers the richness, divergence, or evenness. We use the Simpson's Biodiversity Index to determine the community biodiversity richness. In their survey, the representatives of the various backyards were tested with determining the diversity of the different species, such as urban, suburban, and rural. In addition, the species abundance varies based on the backyards, such as plants, insects, cats, fishes, and hamsters. According to our research, Kajang has the greatest biodiversity of the plant species compared to the other three study areas. However, the lowest index value in the decal, which means the biodiversity is also low. Similarly, the cat species diversity in the Barans and Alosta are both have the same biodiversity. Next. The findings of this experiment will allow us to discover the biodiversity of an ecosystem and find out how the biodiversity helps to keep ecosystem peace-free and stable. That's all from the lab practical sake. I will then pass to the next presenter. Thank you.
Assalamualaikum. My name is NURUL and I will present about our last practical, which is home waste segregation activities. Next slide. How we do that? Firstly, we collect waste from each other's houses and separate that waste according to three classes, which is landfill, recycle, and compost. And list all waste that has been segregated and weight is recorded. Next slide. Okay, for result, this table for a summary of the accumulated waste weight within 2 days. This show that each person produced a different amount of garbage, possibly on the factor of a living area and how many people live in one house. And for discussion, and 3 different types of solid waste disposal method, which is waste, recycle, and compost. First is landfill, which is primarily full by solid waste. It includes garbage from markets, hotel, and resident. And second is recycle, which is collection and breakdown of waste material in order to create something new from that waste, like paper cups, straws, and glass. And lastly is composting, a simplistic solution to the waste that our environment generates, and it also has numerous benefits for both ourselves and the environment. And for the conclusion, they are different in schedule and the way of management of collection weight in every household. It all depends on the resident area. However, among those three, recycling is the most suggested way, as is the most favorable option. That's all for me, and thank you very much.